Okay, well, thank you very much for being here for this talk. My name is Dan Wright. I'm a third-year graduate student in the EEB department, and I'm in the Bernardi lab. And here today, I'm going to talk to you about a phenomenon called genetic cryptic speciation and its impact on species richness in South Africa's rocky intertidal. So I want to start the talk by giving you a description of cryptic speciation. So here we have two species of fish through time as you move from top to bottom. And these fish are defined using what we call the morphological concept of species. So basically, two things look different, we're going to consider them different species. And under the typical speciation scenario, what will happen is you'll have some sort of break in time in the gene flow of a population, and that will generate over time a new species. And that species typically has morphological changes, physical changes, that make it easy to identify. So here, if you did some sort of survey of fish, you're going to say, well, we have 3 species of fish. But what can happen is you can have the same breakdown in gene flow, but no associated morphological change. And so what happens there is you end up with what we call a phylogenetic species. It is genetically diverged, but morphologically, you can't tell the difference. And you would end up with different numbers here. If you surveyed this using the morphological definition, you'd come up with 3. With the phylogenetic definition, you'd come up with 4. So today, I want to share with you a real-world example of this. So I present to you Murinoclinus dorsalis, otherwise known as the white striped-nosed clipfish. It's endemic to South Africa. It lives exclusively in tide pools that you can find at low tide. It likes to live under rocks and make it very hard to catch. And it's those tiny little things, so about 7 centimeters long. And the results that you're looking at, the figure on the top part, is what we call a structure plot. So each individual bar is an individual fish. And
sorry, Daniel, I am unable to see the presentation. Is everyone else able to see the presentation?
Oh.
I can see it.
Okay, maybe it's just me. I don't know why I'm not able to. That's fine. Go ahead.
Oh, shoot. I'm so sorry about that.
No, that's okay. Go ahead.
Well, anyways, in the short period of time, yeah, sorry. To carry on, what the structure is doing is it's separating the individuals into genetic populations. So regardless of where you sampled them geographically, it only cares about how closely related they are to each other when you look at the sequences. And you can see pretty clearly here, on the left you have orange, on the right you have blue. And there's pretty much no individual that has overlap, just one tiny little bit. So it's separating these into distinct clades. And I created this using nuclear DNA, so markers that were from the nucleus of the cell. The bottom portion is a phylogenetic tree that was put together using mitochondrial DNA. So here we were looking at organelles outside of the nucleus of the cell. And the numbers, the names across the middle are the names of the individuals. And they align, so the one bar corresponds to an individual, which is part of that tree. And what's important here, if you guys are able to see my mouse, is right here is the break between the 2 clades in the mitochondrial tree, and that perfectly aligns with the break between the orange and the blue for the nuclear DNA markers. So we have 2 sort of approaches, which is telling us the same thing, which is that this one species morphologically is 2 phylogenetic species. In space, this is what this looks like. So this is the region where I took the samples. It's the western cape of the southern portion of South Africa. And the 2 areas I want to draw your attention are Muli Point and Sea Point up in the top left. So these 2 areas up here are only tens of meters apart. They're effectively one location. But as you can see, the colors correspond. So these are the orange individuals and these are the blue individuals from the plot. And then also Gansbaai down here, you can see that they occur in the same location. So what I'm showing you here is that geographic distance doesn't explain why we're seeing 2 different species. It's not responsible for that gene flow, or that lack of gene flow rather. The implications for this study, there's a couple big ones. What you're looking at here is a map of MPAs in South Africa, but I want to draw your attention to the red square. In the west coast there are very few, there's really only one in the northern region that actually protects any of the coastal area. And this being an intertidal species needs protection in the coastal area. It's relatively common, but in the West, it's under threat from diamond mines and oil mining. And decisions about MPA are almost exclusively made based on species richness. And so what we're saying here is your species richness counts are undercounted, are underrepresented, because you've only used this morphological description. And then the other thing there is any study that is looking at a species, we recommend that you do at least cursory genetic testing, because variation that you see may in fact be because you think you have one species, but you have more than one species, rather than it being responsible or a reaction to some sort of environmental cue or something else. So we think this idea of genetic cryptic speciation is actually relatively common. None of these things happen in a vacuum, so I'd like to thank EEB, Sophie van der Hayden, who is my supervisor down at Stellenbosch University, and Giacomo Bernardi and the entire Bernardi lab. And I will leave you with a picture of my beloved Cape Town and any questions that you might have.
Well, Dan,
yeah.
What is MPA?
Sorry, that's awful of me. Marine protected area.
Okay.
So it's, the marine equivalent of a state park or something like that.
Okay. You can go back to the picture of Cape Town. It was beautiful.
So, Dan, how did you get into this research?
Yeah, I was explaining a little bit before everybody.
Oh, sorry.
No, it's just to acknowledge, I did my master's at the University of Cape Town. And I got introduced to these fish then, along with a bunch of I did a whole bunch of intertidal species, including marine invertebrates. We had crabs, we had snails, we had all kinds of fun things. And then when I decided to continue with my PhD here in California, I started doing this analysis, and we came across this cryptic speciation study, and that's sort of taken over everything.